Okay, so we have finished up our discussion of evolution, and we are going to move on to the second part of the course, which will take the vast majority of the rest of our time. And this part of the course is surveying the diversity of life. So before we talk specifically about prokaryotes, I want to talk about some generalities about how living organisms are related to each other. Now, when people were originally thinking about the different types of life, they divided things into just a couple of kingdoms. They said there was the plant kingdom and the animal kingdom. And, this is something we still do, I think, quite a bit. You're playing 20 questions. One of your questions might be, is a plant, animal, or a mineral? Well, this division of everything into being a plant or an animal works for a lot of organisms, but people started to realize that there are some things that don't fall into the categories of being an animal or a plant, and so the number of kingdoms was expanded. Right? The original plant and animal kingdoms, but then people noticed that, well, fungi, for example, fungi feel about moving around similar to how plants feel about it. That is to say, they're not very excited about it. But on the other hand, fungi don't do photosynthesis, so are they plants? Not really. There's also a produce kingdom, and then the kingdom Monera was created to put prokaryotic organisms in. The five kingdom system was used for a while, but eventually somebody suggested that really the five kingdom system is kind of asking the wrong question. That we need to add in an entirely new level of classification above the kingdom called the domain. So the three domains of life divide all living organisms into the domain bacteria, the domain archaea, and the domain eukarya. There are some interesting results of this change. One of the biggest things that you might notice here is that the animals and plants and fungi are all over here in the domain eukarya. And so while we originally divided everything into plants or animals, well, at the domain level, plants and animals are in the same domain, and these other two domains are not even that closely related to what we thought everything was being a plant or an animal. If we take this phylogeny and we could kind of change it a little bit, so I just kind of simplified it here. So here are three domains: domain eukarya, domain archaea, domain bacteria. And of these three, we are going to consider these two, the domain archaea and the domain bacteria, to be prokaryotes. Now, notice here that the closest relative of the domain archaea are the eukaryotes. And so when we say prokaryotes and include these two domains, well, that's not a monophyletic grouping, right? That is not monophyletic. We're going to use the term prokaryote. We're going to know what it means. Prokaryotes are these organisms that are relatively simple compared to eukaryotes. They have a single circular DNA molecule. They are all single-celled. They don't have membrane-bound organelles. They don't have histone proteins associated with their DNA. But even though they share all those characteristics, archaeans are actually more closely related to eukaryotes than archaeans are to bacteria. So we're going to use the word prokaryote. We're going to know what it means, but we're going to know it's not going to be a phylogenetically accurate term. It is not a monophyletic grouping, but it is useful to know who is related to whom. Not who's related to whom. It's useful to know what characteristics these groups have. In order to know who is related to whom, we have to look at a phylogeny like this to see where the branching actually occurs. Prokaryotes are pretty spectacular because they are the most abundant organisms on Earth. One of the ways that we measure abundance of organisms is something called biomass. Biomass is basically if you take a dead organism and let it dry out, how much is left over, what's the mass of the remaining part. It's estimated that the biomass of prokaryotes is 10 times that of eukaryotes. So in other words, if you took all the prokaryotes, that is to say all the bacteria, all the archaeans, and put them in a pile on one side. And on the other side, you made a pile of all the people and whales, trees, puppies, sharks, mushrooms, and all the other eukaryotes. That the pile of prokaryotes would be 10 times the size of the eukaryotic pile. Right? Being single-celled, we tend not to think about them that much, but holy cow, there's a lot of them. It's estimated that if you pick up a handful of soil, you're going to find more prokaryotes than people that have ever lived. I mean, the current population on Earth of humans, right, is between 7 and 8 billion. And yet a single handful of soil contains more individual prokaryotes than the people that have ever lived. We cannot overstate the importance of prokaryotes. Life as we know it would certainly not be possible without prokaryotes. They are incredibly important. Now, you're going to hear me say that again about other groups of living organisms, and it's going to be true every time. We absolutely need other living organisms, including prokaryotes, for our life. Prokaryotes are found everywhere on Earth. Now, I know sometimes you talk about like humans are found everywhere on Earth, but not really. Prokaryotes are found everywhere. Even in places that are incredibly extreme. Places that would, kill a person immediately. Prokaryotes are found there. The deepest oceans, the tops of mountains, in the air, in the soil, everywhere you go, you find prokaryotes. Now, let me mention that when we say extreme here, we usually use that term in regards to extreme for what a human is used to, right? Keep in mind that some of these extreme conditions we'll talk about are just the normal places that these prokaryotes live. To us, they seem extreme. To the prokaryotes, it's just everyday life. To some of these prokaryotes, where we live seems extreme, and they wouldn't be able to survive here. So I'm just going to give you, these are just some examples of the sorts of craziness that prokaryotes can live within and can put up with. For example, there's a genus called Halobacterium. Halo refers to salt. That's because Halobacterium can live in water that is 10 times the salt concentration of seawater. So I don't know if you've ever been swimming in the ocean and got a mouthful of seawater. It is really salty. These things live in water that's 10 times saltier than that. Another example of extremeness, there's a species called Deinococcus radiodurans. Deinococcus can survive 500,000 rads of radiation. Now, if you're like me, you probably don't own a Geiger counter and know how much radiation that is. For comparison, about 500 rads of radiation would kill a person. So I have a question for you. How is it that Deinococcus was able to adapt to survive such incredibly high levels of radiation, even if there's nowhere on Earth that you'd find a radiation close to that? How do they adapt to this thing that they would never experience? Well, you might say, hey, maybe they came from space. But we don't think so. We think that they live here and have always lived here. Well, it turns out that Deinococcus has probably adapted for something else. And that adapting for something else has given it a very robust ability to avoid being killed by radiation. Specifically, Deinococcus lives in areas where it dries out pretty often. And one of the dangers of drying out is that DNA becomes more brittle and more likely to break. So Deinococcus has very stable DNA. Sort of stable DNA that could put up with things like radiation that might normally damage the DNA. Deinococcus has DNA that can survive that. And so it is adapted for one thing, that is to say, avoiding being damaged by drying out, and yet uses that for something else. Does that sound familiar? Did we talk about anything else like that? Maybe we talked about something using a tire iron to hit somebody in the face when they try to steal your car. This would be another example of exaptation. Right? Something that evolved for one purpose, but turns out to be beneficial for something else. So Dinococcus can survive ridiculous radiation, but that's not really what it adapted to do. But it turns out it's helpful when that happens. All right, let's look at another really ridiculous example. Picrophilus can live in water that, although I don't even know if you call it water at this point, that has a pH below 0. Right? So if you remember the pH scale, it's a logarithmic scale. So that means that a pH of 6 is 10 times more acidic than a pH of 7, and a pH of 5 is 100 times more acidic than a pH of 7. Picrophilus can live in a solution that is below 0, that is ridiculously acidic. Right? Stomach acid is between 1 and 2 in the pH scale. This is a pH that's outside of what we normally even think about. I mean, you usually think about a pH scale between 0 and 14, but it turns out the pH can be negative. And when it is really, really, really acidic. Crazy, right? Things that a eukaryote would never even think about surviving. Some prokaryotes are like, yeah, no problem. That's where I live. We think that prokaryotes were probably the first organisms around 3.5 billion years ago. They may have developed. We have some microbial mats on the floor of the ocean that are probably pretty similar to those first organisms. We also have these structures called stromatolites, which are basically these little pedestals of prokaryotes. And how it works is the lower down prokaryotes, as they're growing, they die, and their sort of bodies result in these like layers. And so you can see in the picture on the right, here are these individual layers that have built up over time and created these stromatolites. The top layer, such as the picture on the left, might still be alive, but everything below it is sort of leftover remains of prokaryotes, and these layer after layer after layer after layer. Being, again, single-celled organisms, you have to remember there's going to be a lot of individuals, a lot of layers to be these pedestals. We think that prokaryotes were really important in the history of life on Earth for a variety of reasons. One of these has to do with atmospheric oxygen. So you might remember the early atmosphere is very different than what we have now. At some point, it's going to switch over to an oxygenated atmosphere like we have today. Our current atmosphere is somewhere around 20-21% of oxygen. We think that that source of that oxygen, that switch from no oxygen to oxygen, came about from biological means, specifically from organisms that were able to do photosynthesis. One of the byproducts of photosynthesis is oxygen. The group of prokaryotes we think that first oxygenated the atmosphere is a group known as the cyanobacteria. You might hear these referred to as blue-green algae. We're going to stick with cyanobacteria. We're going to try to use the word algae, only refer to eukaryotic photosynthesizers. So we're going to stick with cyanobacteria for this course. Cyanobacteria, you can see in the picture on the right, there's this kind of greenish tint to this water. It's because there are cyanobacteria, these prokaryotic organisms that are living there, doing photosynthesis, producing oxygen. That oxygen becomes important for a few different reasons, one of which is that oxygen can then serve as the final electron acceptor during cellular respiration. That presence of that oxygen means that organisms like us that do aerobic respiration are able to harvest a lot more energy from our food than if we didn't have access to that oxygen. That efficient metabolism means we can have large organisms that move quickly that would not be possible if that oxygen was not present. Another benefit is that oxygenated atmosphere resulted in the development of ozone, whose chemical formula is O3, 3 oxygens, and that ozone serves as a shield for ultraviolet radiation to keep organisms on the surface of the Earth safer from the damaging sunlight, the damaging UV radiation that's in sunlight. So both of these things are really key to the success of organisms, including humans here on Earth. Now, it's also important to realize that just because a lot of things benefited doesn't mean there weren't things that suffered a detriment from this. There are a group of organisms known as obligate anaerobes that cannot survive in the presence of oxygen. We still have them. They're very limited in where they can live now. Prior to the oxygenation of the atmosphere, they could have been anywhere. But now they have to live in places where they can hide from the oxygen. And so a lot of species probably became extinct because there was no place left for them to be away from that oxygen. Another way that prokaryotes are important has to do with nitrogen. There are some that are able to fix nitrogen. By fix here, we don't mean that the nitrogen is broken. We mean that nitrogen that's in the air, right now we're breathing in nitrogen, it's in the form of N2, two nitrogen atoms attached together, changing that into a usable form, usually into a form something like ammonia, a form that living organisms can take advantage of. Nitrogen gas, like you're breathing in right now, that makes up the majority of the atmosphere, that is two nitrogen atoms held together with a triple bond. And most organisms can't deal with that triple bond, can't break that apart to get access to that nitrogen. Nitrogen is incredibly important, right? It's used in like pretty much everything in a living organism, right? Amino acids have nitrogen in them. DNA has nitrogen in it. Nitrogen is really key, but yet most of the nitrogen we come in contact with we can't use because it's in that N2 form, that gaseous form of nitrogen that most living organisms can't take advantage of. The only living organisms that can fix nitrogen, that can take it from that inorganic form and put it into a usable form, are prokaryotes. The picture there on the right, actually, this shows the cells of a cyanobacterium. And some of those different cells there, those heterocysts are able to fix nitrogen. Now, I will mention there is one other way nitrogen gets fixed aside from living organisms, and that is lightning. As lightning streams down through the atmosphere, it breaks some bonds between those nitrogens, and some nitrogen is fixed that way. But something like 90% happens biologically through prokaryotes, because prokaryotes are the only organisms that can fix nitrogen. Prokaryotes are also really important when it comes to carbon and energy. So we'll talk about where organisms kind of in general get this, and then we'll think about how prokaryotes do it. So some organisms are able to get energy from sunlight. We're going to call those phototrophs. And then some organisms are getting their energy from chemotrophs. So we as humans, since we can't capture sunlight energy, we're chemotrophs. Sources of carbon include those that are able to fix inorganic carbon, so to take carbon from something like carbon dioxide and use that as a carbon source. Those are autotrophs, whereas heterotrophs, hetero meaning different or other, have to get their carbon from other living organisms. So when you eat food, you're getting organic carbon, carbon that has already been fixed by an autotroph. Now, we can also then do combinations of these. So we could say, let's say a photoautotroph gets its energy from sunlight and is able to fix inorganic carbon. So something like a plant doing photosynthesis is a photoautotroph. We as humans, we are chemoheterotrophs, right? We get our food and our energy from other organisms. Now, it turns out that prokaryotes can do every combination. They can be photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. They are the only group of organisms that have such a diversity in their energy and their carbon sources. There are prokaryotes that pretty much do everything. They live everywhere, they can survive everything, and they do everything. It's a little bit of an exaggeration, but not too much. All right, let's talk about the structure of prokaryotes. So prokaryotes are single-celled organisms. There are some that exist in colonies. So colony means groups of single cells basically living together. The cells of prokaryotes are pretty small, usually 1/2 to 5 micrometers. Just to give you an idea, the average human hair is something like 100 micrometers in width. And so a large prokaryotic cell is about 1/20th the diameter of human hair. So pretty small, pretty small. There is an exception to that. There's this weird thing, Thiomargarita namibiensis. Thiomargarita is actually pretty big. You can see the scale bar there says 1 millimeter. So this is big enough you can see it with the naked eye. Thiomargarita kind of cheats though. It's basically like a balloon. The cellular material is all on the kind of the outer surface of the balloon, and the middle part is kind of empty. So there's just a thin layer of living part around the outside, and the rest of it's just empty. And so that's kind of how it cheats to get to be large. It's like a regular prokaryotic cell that you inflated. Thiomargarita namibiensis. Thio means sulfur. Margarita refers to pearl. So it's a type of bacteria that does stuff with sulfur. Looks like a string of pearls. And it was probably discovered in Namibia, is where the namibiensis comes from. Let's look at the shapes of prokaryotes. So we're going to divide them into 3 different cellular shapes. Far left, the coccus shape is that spherical shape. In the middle, we have the bacilli, which are kind of rod-shaped or sausage-shaped, sometimes they're called. And then on the far right, we have the sporulli. The sporulli are the spiral-shaped, or I like to think of them as the pasta-shaped. Something like rotini. So the 3 basic shapes that they come in are the cocci, the bacilli, and the sporulli. Let's talk about those colonies in which they exist. So we generally divide them, those that don't live by themselves, those that are in colonies, into pairs, chains, or clumps. So here's pairs, and we could refer to those with the prefix diplo. Here are chains, we're going to call strepto, and then clumps, we're going to call staphylo. Now, what we can do is we can take that prefix, like diplo, and we can combine it with the cell shape. So on the far left, we have diplococcus. So diplococcus, excuse me, I had to sneeze. Diplococcus are these spherical cells in pairs. In the middle, we have spherical cells in chains. We're going to call those streptococcus. Far right, spherical cells in clumps. We're going to call those staphylococcus. Now, I want to point out that the words we have here are just describing the cell shape and the colony arrangement. Streptococcus, as I have written on the screen there, you might notice, is not italicized. That means I'm not referring necessarily to the genus Streptococcus. Yes, strep throat is caused by an organism in the genus Streptococcus, but it's possible for a prokaryote to be a Streptococcus, that is to say have spherical cells in a chain, but not be Streptococcus, that is to say the genus Streptococcus. Right? Same thing, Staphylococcus. Yes, it's a genus, but it's also just a description. Right? So if it's not italicized, then it must not be the genus, it must just be a description of those cells. Let's talk about the what I'm going to call the living arrangements of prokaryotes. Many are free living. Free living meaning they're just kind of out by themselves hanging around. But there are some that will join in to biofilms. And a biofilm is basically when a bunch of prokaryotes get together and exude this sticky polysaccharide matrix, basically this glue that glues them together. And it can be multiple different species of prokaryotes. It could even be bacteria and archaeans both joined together into this biofilm. Now, you might be familiar with this. You know, if maybe you forget to brush your teeth for like a week, and then you touch them and you're like, hey, it kind of feels like there's like carpet on my teeth. That is a biofilm. That plaque is a biofilm. That's a bunch of prokaryotes that have started living together that have secreted this glue that holds them together and puts them on your teeth, for example, and makes it harder to get them off of there. Now, fortunately, our teeth, we can just brush our teeth because biofilms are pretty good at withstanding antibiotics. And so when a biofilm forms someplace where it's tougher to get to, let's say on heart valves, for example, well, it makes it a lot more difficult to get rid of that biofilm because we can't just reach in there with a brush as easily as we can brush our teeth. I think biofilms are kind of interesting because it's kind of interesting to think about how they first form, right? About how these prokaryotes have to communicate with each other and decide, hey, it's time for us to do this same thing. I don't think about prokaryotes as being all that, I don't know, communicative, talking to each other and discussing things and deciding if there's enough of them around if they want to do it. But they do, and then they form these biofilms. Once the biofilm is formed, then they can kind of reproduce. They can release cells out into the world, which can go find someplace new to grow another biofilm. So, communication, coordination, decision-making, things I don't think about with prokaryotes, but things that they're able to do. All right, let's look at the cellular structure of a prokaryote. So I'll remind you, as I mentioned before, they are simpler than eukaryotic cells, although don't make the mistake and think that they're simple. So if we take a look, there are some things that we're going to find in most, if not all, prokaryotes. So DNA, we're going to put that in an area known as the nucleoid. So there's no membrane around the DNA, so it's just a region we'll call the nucleoid. Single circular DNA molecule. There's a membrane around the cell. Most prokaryotes have a cell wall. Sometimes there's this sticky structure known as a capsule. There might be projections known as pili. Ribosomes. What do ribosomes do? That's right. Ribosomes make protein. Ribosomes are present. And then some have a flagellum that they can use to move. So not a lot of parts to the structure, not as much as we see in a eukaryote, but don't get misled into thinking that they're simple. Just because they're simpler doesn't mean they're simple. They're able to carry out all the functions of life in that single cell. Let's talk about those structures that they have on the cell surface. I mentioned this capsule. It's kind of like a sticky, almost like a glue layer on the surface of the prokaryote. Provides the benefit of letting them stick in place. So if there's some place where they don't want to leave, they can kind of glue themselves down. It also helps protect them from drying out and also protects them from being attacked by the immune system. I mean, I guess you can kind of see why, right? If you covered yourself in glue, probably nobody would mess with you. Same sort of thing with the prokaryotes. The immune system has a tougher time attacking them if they're covered in this thick layer of stickiness. The flagella is used for movement. About half of prokaryotes are able to move, so they can use that to propel themselves. So not all of them can, but those that can do so with a flagellum. They also have these pili, which are these kind of hair-like structures coming off of them, useful for attaching to surfaces, for holding them in place.